This is in the way. All right, can you guys hear me okay? I can. Let me close my door real quick. I'll be right back.
Hello.
Hey, really close on my face. Why is it this close? There it goes. What do I want to share? Screen 4, I think. Sure. Let's see what it's showing. Yes, that's what I want to see. Actually, share this screen. Sorry, I had a meeting right before this, so I am kind of just trying to make sure I'm oriented. Screen 3. Sure.
We had to wrangle a 2-year-old.
Yeah, must be fun. Okay, good, good. I'm happy with that. Maybe I'm not happy with that. I also have a new computer set up, so me trying to figure out which computer screen is which. I have 4 screens now, so just bear with me here. This is the screen I want. Okay. Drag this over to here and open up my chat in case you guys chat. And my participants. There we go. That's better. Okay, so let's get started. Sounds like fun. Yes, okay. Super weird having all these screens open. Okay, so we are going to be talking about chapter 17, which is our disorders of the immune system, and then chapter 18, which is our diagnostic test methods. It really shouldn't take us super long to cover all this stuff. I really don't think it'll be that bad. So let's get into it. This is our slide on the concepts that were from chapter 17. Let me see what I want to cover here. First of all is our hygiene hypothesis. That hygiene hypothesis is the idea that lack of exposure to microbes is associated with allergies later in life. So if you didn't have a lot of exposure to microbes early on in your life, then you might be, this is really freaking me out, then you might be at risk for allergies at another part in your own life. This is annoying. There we go. Okay. So just put it in perspective, because it helps to train your immune system basically when you're exposed to a lot of microbes early in life, so that later in life you don't misidentify things basically with your immune response. So that's the idea with that. We have 4 types of hypersensitivities and 2 types of hyposensitivities. So again, hypersensitivities here, we're talking about the overreaction of the immune response to things. And this is often relating to reacting to something that's not a threat, but your body perceives it as a threat. And so there's 4 different ways that your body could do that. The first option is our type 1 response. So type 1 response is governed by IgE. You've made an IgE, which is a type of antibody. Your body got signals to make IgE to something, and then the IgE basically binds on the surface of mast cells. I don't know why I drew it so small, but I did. Mast cells, they will basically wear the IgE like little flags, and then whenever they see antigen come and bind onto them, then when the antigen binds to them, and again, in this case, the antigen is the allergen, right? Then that will cause the mast cells to degranulate, generally speaking. Okay. So that's the idea there. So that's the type 1. This could be caused by things that are inhaled, injected, contacted, like on your skin, or ingested, so eaten. So those are all things that can lead to development of allergies that are mediated by IgE, leading to degranulation of the mast cells. You have to have an initial sensitization, meaning, just a little mini review here, you have to see the antigen and recognize that antigen has to be eaten up by a macrophage, which then presents it on MHC2, where it is recognized by the T cell receptor on a CD4 T helper cell with the help of CD4, right, which would lead to activation, leading to a lot of T helper cells, some of which will be Th1 and Th2 effector helper cells. The Th2s can then send the signal to B cells that have immunoglobulin on their surface in the form of IgD. Then they'll see that, they'll bring it in, put it on MHC2. Bam. T cell receptor on our Th2. CD4. Then we activate that B cell. Then that B cell becomes a plasma cell that then cranks out first IgM antibody, and then we will class switch to IgE. Then that IgE can be worn by the mast cells. So all of this, all of what I just went through, is that initial sensitization process. We have to sensitize first. We have to have our initial reaction with our antigen, which in this case is whatever your allergen is, in order to trigger making the IgE that the mast cells wear. Then they contact the antigen and degranulate. Okay, so that whole idea is basically what that desensitization process is. So that's excuse me, sensitization. Sensitize. Again, just like we talked about in chapter 16, this is your primary interaction with the antigen. Any response after that will be your secondary or amnesiatic response, or amnestic, however you want to look at that term. It means the same thing though, okay? All right, so again, IgE on the mast cell sees the antigen the second time, and now we can degranulate, and that'll happen every time that you see that antigen. So basically allergies reacting to an allergen can be a local response or a systemic response. If it's local, if it's in the upper respiratory system, eyes and nose and the sinuses, anything in those general areas together, that's still local, that would be atopic. But if it's throughout your bloodstream and spread throughout your body, we call that anaphylactic. And that response, an anaphylactic response, can lead to basically like shock in the body, and that can lead to death of the person within a very short period of time as a result of that. All right. How do we treat this type of response? Antihistamines are pretty common for atopic types of responses. Steroids are effective for atopic type responses, but for anaphylactic response, we want to treat with epinephrine. Epinephrine is the medically delivered version of adrenaline. Okay, so that's the primary way that we would treat this. You can also instigate adrenaline through other ways, but adrenaline is made by the body, epinephrine is delivered medically. We can train the body that basically when we make IgE, we can re-expose the body to that antigen and train it instead to make IgG. If you can class switch to IgG, you'll still produce the IgE, but you'll make IgG, you'll make more of it. And then the IgG will bind up with the allergen and bind it all up so that it can't interact, doesn't get the chance to interact with IgE. Yes, okay. So that's the idea of how to basically control the type 1 responses. All right, so I'm going to go ahead and erase all this. Just do this. There we go. All right, so type 2 responses. Get a little bit closer in here so I can draw. All right, type 2 responses. Type 2 is governed by basically IgG or IgM plus, well, leading to complement lysis via what we call the membrane attack complex. We talked about this in chapter 15. This is where the parts of complement, after we like set off the bomb system that is complement, that once when we do that, we can take the parts of that and build a big hole in the target cells. And in this case, whatever the antibody is bound to. That's useful when the antibody is bound to a pathogen. When the antibody is bound to blood cells, then you're lysing blood cells open. And so that's what happens in a type 2 response. In type 2, it's usually going to be blood cells. Now, in other autoimmune disorders, we can target other cells as well. It doesn't have to be blood cells, but it's primarily talked about in our blood rejection type of situation. So we have IgG or IgM binding to blood cells and then initiating the complement cascade. Remember that that pathway of initiating complement cascade from antibody is known as the classical pathway of complement activation. That will lead to lysis of the target red blood cell. So if my body sees type A blood as self, but it sees type B blood as foreign, it'll make IgG or IgM to the B molecule on B blood cells and then lyse them through the classical pathway of complement. Okay, and that will basically burst open those B blood cells. And then they will leak their contents and my spleen will react to it and my body will try to clean it up and get all these signals of red blood cells bursting open. And that can lead to shock and death. The most common outcome of getting transfused with the wrong blood type is going to be death. Here we also have to understand that if you have genes for A or if you have genes for B, you will express them. Period. Equal. Okay, so if you have a gene for A and a gene for B, one from your mom and one from your dad, you will have AB blood. If you have a gene for A and a gene for O, you will have A blood because A and B will be expressed. If you have a gene for O and a gene for O, that's the only time that you will get type O because it's recessive. You need 2 copies of it for it to be expressed. Okay. What else? And we go over this in quite a lot of detail in our lecture recordings if you didn't watch that. The other side of blood isn't just the ABO groups. So we can think of blood basically as having a base that represents the O blood type. And then if you have the A, then you're an A. This is what B would look like. And O would look like that. So if you're AB, you have both the A and the B. But to everybody, that base looks like self. So anybody can get O, right? So here's some other examples we could talk about. A, it can get O and A. B, it can get B and O. O can get O. AB can get A, B, AB, or O. Okay, so that's the general breakdown of how that's going to work. The other side to this isn't just the ABO group, also the RH group. RH group is also known as D antigen, commonly called either one. Okay, so if you are RH positive, then we would call you A positive or B positive or AB positive. Okay, that's the positive onto it. If you're RH negative, then we would call you A negative or whatever. So the idea with that too is that there is a base associated with this as well. Again, you have your red blood cells and you have your base for your ABO group, whatever you are, A, B, or O. If you're O, you just lack an A or a B. You also have another base. If you have the D antigen, then you are Rh positive. If you don't have the D antigen, then you are Rh negative, but you'd still have the base. Okay. All right. And so this basically determines Rh positive or Rh negative in our blood types. The idea here is that if you're Rh negative, anybody, positives or negatives, can get Rh negative, because positives see Rh negative as self. They see Rh positive as self, but an Rh negative sees Rh positive as foreign. That's the idea with that. So now we don't just have to worry about ABO groups, we also have to worry about Rh groups in that regard. So with the presence of that. And so this also gets into that idea of that hemolytic disease of the newborn. The mama can attack the baby. The mama can make antibodies to the baby. So even though you might be an O negative mama, here's our mama, and here's her belly where the baby's growing. If the baby is O positive, but the mama is O negative, she's not attacking the ABO group, right? It's the same ABO group. That doesn't look weird to her. But this positive looks weird. So she will make antibodies, IgM or IgG, against the baby's positive group. This isn't a problem because that placental barrier will keep that in check until the baby is born. Once labor initiates, the antibodies will attack the baby. And so this can lead to the baby dying either during birth or shortly after birth. It's not an uncommon outcome for that. So that's called hemolytic disease of the newborn. All right, moving on. Get all of this, delete that. Okay. Next thing is the type 3 immune complex. So we did type 2. That's not what I'm trying to do. We did type 2, type 1. So we're now on type 3. Type 3, you have antibodies, and they're hanging out, and they find their way to bind to antigen. So here's our antigen. So these guys will bind up to antigen, and we'll get antibodies that'll bind onto it and create these complexes and whatever. This is the idea of the immune complex here, all right? Then after we create these complexes of antibody and antigen, and we'll create this big old complex of antibody and antigen. This can either get eliminated by the body naturally, or they can get stuck in tight spaces in the body. And this includes the basement membranes, which is basically like surfaces in the tight areas of the body, including in capillaries, in the heart and lungs, where we have a lot of gas exchange happening, in joints and in the kidneys, where we have a lot of basically electrolyte waste and gas exchange going on in the kidneys as well. And so these immune complexes will get stuck. This couldn't be making my face look redder if I tried. These immune complexes will get stuck in those basement membranes in the tight spaces of the body, and then lead to neutrophils getting excited because they'll come and see the antigen and try to eat it up and send out all these inflammatory cytokines in those tight spaces. We see this associated with a lot of autoimmune disorders, but one that is associated with infection is acute post-streptococcal glomerulonephritis, which is quite a mouthful. In this, you have a streptococcal infection, like strep throat, okay? And your body creates antibodies against the bacteria. Some of the antigens of the bacteria get caught up with antibodies and get trapped in the glomerulus of the kidneys. This is where a lot of the exchange happens of like wastes out of the bloodstream and then into the urine to get rid of it, to expel it. So this happens a lot in the kidneys. And again, in the structure of the kidneys called the glomerulus. It's a very tight space. And so these complexes get stuck there, and then you have the inflammation of the neutrophils in those tight spaces in the kidneys. And then that will lead to degranulation of those neutrophils and damage of the kidney tissue around the area as a result of trying to clear out antigen from strep. So it's acute because it usually doesn't last very long. Usually as long as the antigen is around, until the antigen gets eliminated, this could be causing damage to the kidneys. But once the antigen is eliminated, this won't continue. However, we could see this happening if we get immune complexes associated with self-antigen, that this could happen. And so this happens a lot in the tight spaces in the body in our autoimmunities. Whenever you're not going to get rid of the antigen because it's self. You're always around. As long as you're alive, you have self-antigen to instigate the immune complexes. So in that case, the immune antigen can continue to cause inflammation chronically in autoimmunities. And so we see those autoimmunity situations happening a lot with SLE, which is systemic lupus erythematosus. Lupus is what we just call it typically. That happens in the capillaries of the face. In people who have lupus, it leads to a butterfly rash. In rheumatoid arthritis, whenever we have swelling of the joints, and then the joints end up getting swollen from that inflammation from those immune complexes there. So that's a common one of type 3 as well. Cool. Last one for our hypersensitivities is going to be type 4. So we get type 3. Now we're on type 4. Type 4 is mediated by the T cell response, and specifically our CD8, like our inside response. Okay, so when we activate our CD8 T cells, again, you will have an infected lung cell or something, an infected cell, asking for help from something inside. And this will happen on MHC1. Then the T cell receptor, if this is MHC1, this T cell receptor is on a cytotoxic T cell. And we recognize that it's MHC1 with that CD8 molecule. Then we also have to get that trigger, that go ahead from that TH1, which we activated previously. Okay. Once we get all these signals, we can activate. Then our cytotoxic T cell will release perforins and granzymes. To attack the infected cell and kill it. It can also release things to activate macrophages, which are going to be involved in more inflammation and everything like that. So all of this is happening from this response. And this is what drives our type 4. When we activate our cytotoxic T cell, we also make some cytotoxic T cell memory cells. Okay. We only make a few of them. Well, 2 or 3 cytotoxic T cell memory cells isn't going to be a lot if you see an antigen again in an actual infection situation, which is why antibody is so important, because we can make antibody right away. But we got to wait on our cytotoxic T cell guys. And these won't take as long to multiply out, but they've still got to multiply. And this usually takes anywhere from 24 to 72 hours. So 1 to 3 days. So once these guys start acting up, then we will start seeing that inflammation process from them. So if we're attacking self, or if we're attacking, something else that you could have this type of response to. We see this a lot with contact dermatitis. So poison ivy or certain types of jewelry or metals, you would see this type of an effect. So it will take at least 2 days for this to get re-triggered back out. So that's why when you get poison ivy, you don't get the rash within minutes like you would an allergy response. You get the rash within a couple of days because it's mediated by this delayed type hypersensitivity. This is also what mediates the response to a PPD skin test for tuberculosis. We use tuberculin antigen, put it just under the skin. And if you have seen tuberculosis bacteria before, again, Mycobacterium tuberculosis, then you'll have T cell memory that would be reactivated and then come out and attack your skin as a result. And if that happens within a couple days, then you've seen tuberculosis before, you have memory of it, and you probably have it. It could be latent, or you've had the immunization for it, which is not commonly given in the United States. This also is what determines graft rejection. So hold on really quick while I delete this stuff. I didn't get everything. I don't know why it's not letting me delete everything. There we go. All right, so in the case of graft rejection, if my MHC1 on all of my cells, all nucleated cells, all my cells look like this. And all of your cells look like this. They have MHC1 also. How do I want to draw this? MHC1. I don't know. Got a different shape to them. My CD8 molecule on my cytotoxic T cell with its little T cell receptor. Okay. And let's say my T cell receptor matches up with your MHC. Well, my T cell receptor might be able to see your MHC as foreign and still give the go-ahead with the CD8 and attack your kidney. This is actually pretty common because remember, you will have a T cell receptor for 8.6 billion different antigens, and they'll stay active and be able to react to any antigen ever. And unless we train it to see it as self. And I've never seen your cells. I never got told that I should see that as self. So if you put one of your kidneys in me, my body will kill all of your cells that were in your kidney, unless your MHC looks exactly like mine. So genetic testing is done to make sure that the MHC looks the same. In that type of situation where you're getting a graft from one person to another person, and we need to make sure that their MHC matches, we actually call the MHC HLA, which we call human lymphocyte antigen. So it's basically like human leukocytic antigen, but it's basically an antigen that could be seen by my white blood cells as foreign. We want to make sure that I see yours as self. So we do a lot of genetic testing to try to predict for that. It's not perfect, but it's the best that we have. Because 8.6 billion different antigens, man. I mean, I could easily see your kidneys as foreign and then reject them. And that's what governs organ rejection. For the different types of grafts that you could get. So the different types of grafts you could get could be autograft. So that's if it's self to self. Isograft, which is identical twins, which you should have almost no rejection likelihood there. It's unlikely. Allograft, which is from person to person, not identical. Okay. And then xenograft, which is from an animal, like a heart valve from a pig or a cow. This is also a part where we would say to be aware of, what am I trying to say here? Host versus graft and graft versus host. So if I have a kidney from not self, and the host self cells attack the kidney because it looks foreign, then this is the host versus the graft. If you get bone marrow, which one of the precursors of all white blood cells from bone marrow, the hematopoietic stem cell, which begets cells like cytotoxic T cell, which might then attack the host. When the graft produces T cells that attack the host, this is known as graft versus host. So this only happens with bone marrow transplants. If the host's body immune system is attacking the graft, that's host versus graft. If the graft is producing cells that attack the host, then that's graft versus host. Okay. Then we talked a little bit about autoimmunity already. Whoops. This does not want to cooperate. All right, we already talked a little bit about autoimmunity. It just doesn't want to erase, that's fine. And it could be any of these class types. It could be type 1, 2, well, not usually type 1. Type 2, 3, or 4 are usually associated with autoimmunity. And it depends on the immune response and the autoimmunity. Type 1 diabetes, for example, in type 1 diabetes, we have a CD8 response killing insulin-producing cells. That would be a type 4 hypersensitivity leading to autoimmunity that causes type 1 diabetes. So there's that one. So this can be triggered from seeing pathogens that look like self, which is molecular mimicry. You can even have viral infections that alter your own cell's surface to make it look foreign. And that can be a permanent change. And then that could lead to all that training that you did to train your body on what self was, just completely negate it. And now we can start attacking self tissues because it's changed because you had a viral infection. And now it looks different because of that. We can also have changes in the gut biome, and the gut biome is so important for training our immune system. It's probably one of the most important things for training our immune system. So if you have a change in your gut biome, or if you have a very poorly established gut biome early on in life, then that could lead to issues with training what is self and what is non-self and when to react to foreign things. That can lead to autoimmunity. All right, finally, we get to our immunodeficiencies. Primary immunodeficiencies were ones that were present from birth. You have a genetic abnormality that led to you having an immunodeficiency, whereas secondary immunodeficiencies were acquired later on in life. And it could be very early on in life, but it's still, if it's acquired, it wasn't genetically ingrained in you from conception, then anything that's acquired after that. So this could be cancer, like leukemia, where you have overgrowth of a certain white blood cell that is not going to be working properly. So then the properly working ones can't function properly. This could be from AIDS, which is acquired immunodeficiency syndrome as a result of having HIV, because HIV infects your T helper cell captains. And so you can't activate an immune response properly if your captains are dead from being infected with HIV. Just saying. HIV leading to AIDS is basically, AIDS is determined by how many T cells you have, your T cell count. And then once your T cell count drops below a certain threshold, you're significantly more likely to have opportunistic infections that define AIDS. As well as radiation and chemotherapy, often used to treat cancer, but also could be radiation just from environmental exposures, like from X-rays and things like that. Can cause issues with the immune response as well. All right, does anybody have any questions about chapter 17 before we move on? All right, we're going to chapter 18. Chapter 18. So I'm just checking, make sure I'm not missing anything. All right, chapter 18. Most of chapter 18 was dealing with categorizing a lot of the testing methods that we've already talked about, or putting some of the stuff we've already talked about into a certain perspective as far as testing for pathogens. First things first, you have to be aware that you have to collect your specimen. Either that's from swabbing a nose or collecting a fecal sample or a biopsy of tissue or whatever it could be. And then we may need to cultivate the specimen. And depending on if you have a predilection or a prediction on what you think the pathogen could be, that could help you choose how to cultivate the specimens as well, just to reduce contamination from the microbiome, right? So that could be it. And it also means that we have to have perspective on how many microbes should be normally found in an area. If I culture your poop, and I see a few E. coli colonies, I don't think I'd be worried. If I culture your poop and it's like all E. coli, that's a problem. So that puts perspective into that. However, if I look in your lungs and I see even one Mycobacterium acid fast bacterium, that's a problem. There should not be any in there. So depends on the place, depends on the number of organisms, depends on your sample and how you collected it and all of that. You have to have perspective on that stuff. And then we might do testing to determine it, what it is. We talked about that a lot in our lab, the recent lab that we did. So we talked about how to use that dichotomous key to identify our organism. Because imagine you thought your patient has a skin infection and you think, oh, this is staph, easy. And you're like, it's probably staph. Let's go ahead and do PCR just to confirm that. But the PCR comes back negative for staph. Well, now what? Now what do you think it is? Well, you might think, oh, maybe it's strep, and you might do another PCR. What if it's not staph or strep? What now? So that's where all the testing that we're doing in lab comes into play. All the testing that we are doing in lab right now are biochemical tests. These are tests that are often based on color changes, based on products of a biochemical pathway. Does the organism ferment glucose? If it does, it made acid and turned to this yellow. Wasn't the organism able to use citrate as a carbon source? If it did, it used citrate, therefore used the nitrogen source, made ammonia, pH went up and caused it to turn blue. Stuff like that. We can do a whole bunch of these tests like we're doing this whole part of the semester. And we could do them all up front. And then in a couple of days, come back and see that it fermented glucose, it fermented lactose and made gas. It broke down citrates. It made urease. It was catalase positive. And we get all this info together, and now we can use things like dichotomous keys, which there are databases for this stuff that your lab could plug into a database and say, it had all these things positive and all these things negative. Therefore, this was, this other kind of bacteria, Pseudomonas aeruginosa. It wasn't ever staph. It wasn't ever strep. It was always this bacteria from those tests. And we didn't have to keep trying different PCRs to figure that out. So we could use these tests to identify it that way. So that's the idea with this stuff. If it's based on biochemical stuff, like we've been doing all in lab the second 8 weeks, those fall into a category called a phenotypic test. Phenotypic tests include biochemical tests, antimicrobial sensitivity tests, and phage typing. These are all phenotypic. If it depends on the, and I should talk about phage typing. I just blow over it like you even know what it means. Phage typing is referring to the fact that a bacteriophage, which is a virus that infects bacteria, there are no bacteriophages that affect multiple species of bacteria. All bacteriophages only infect one species of bacteria each. So bacteriophage A infects this bacteria. Bacteriophage B infects this species. Bacteriophage C infects another species. And so if your patient has diarrhea, how many freaking types of pathogens could potentially cause diarrhea? And if you all are thinking, well, it's salmonella, then you ain't gotten, you're going to learn. You're going to learn in chapter 23. You're going to learn. There are so many types of pathogens that can cause diarrhea. It is wild. And different kinds of bacteria too, not just salmonella, not just E. coli, not just Shigella, not just Campylobacter, not just Listeria. There are so many, there's just so many. And so if your patient has diarrhea and you're pretty sure it's going to be salmonella, you might use a bacteriophage that infects salmonella. If it infects your patient's poop culture, then your patient has salmonella. That's how phage typing works. It depends if the phage can infect your patient's culture, your patient's pathogen culture. If it didn't infect it, then it's not salmonella, it's something else. And we can do other tests to determine what it is. So your patient has diarrhea. You collect the specimen, the stool sample, and you culture it and infect it with a phage that infects salmonella. And let's say it doesn't work. Your patient sample doesn't get killed by the phage. Your patient doesn't have salmonella. So what now? Maybe you take that culture and grow it on EMB. And let's say it grows on EMB agar and it turns it metallic green. You have E. coli. So we can do all these tests and break it down. And if you're really not sure, you could do all of them at once. And you don't have to waste money on a PCR, multiple PCRs for multiple different types of pathogens. Just take a culture of their poop. Some of it infect with salmonella phage. Some of it do on EMB. Some of it do X, Y, or Z thing. And then you got info and you can move forward. And that's how you might do your diagnosis, doing multiple of these tests at one go. I hope that helps. I hope that helps understand why we do this stuff and why we're doing it in the lab this way. All right, so that's our phenotypic test. Phenotypic, that'll be in our orange. Purple will be immunologic tests. Immunologic. Anything that we do in immunologic tests will be based on antigen recognition. So it could be antigen and antibody. Most of it's antigen antibody. It's either antibody or T cell. But it depends on your immune response or some kind of targeted immune reaction. So identifying antigen or identifying antibody. That's what all immunologic tests rely on. Okay. If we're identifying the antigen, it's a direct test. If we're identifying the antibody, it's an indirect test. Okay. All right, here are some examples. Immunochromatography. You have a piece of paper and an antibody with an enzyme on it. You put the patient's pee on the paper or nostril fluid that you got or whatever. You put the patient's sample on the paper and it runs through the paper. If the sample hits it and causes that line where the antibody is to change shape, because remember, antibody to enzyme, enzyme cleaves a thing and causes a color change that you can now see on the paper. It works the exact same way as ELISA, but it's like on a filter paper. That's immunochromatography. Chroma is referring to the color change there. All right. Antibody titer, measuring the number of antibodies in your patient's sample. Okay. What else do I want to say? In vitro serum testing. There's a variety of in vitro serum testing that we could do. One that you're familiar with is ELISA. Another one that's similar to ELISA is immunofluorescence. In immunofluorescence, we would have a sample that's on a slide and add antibodies to it. And that antibody is linked to a fluorescent marker that's like a color. If the antibody sticks, your patient has the antigen. Immunofluorescence. The antibody sticks, the patient has the antigen. And how would you see the antibody sticking in this case? Because now your sample glows a color. This is not related to pea glow. This is not related to pea glow. This is not related to pea glow. This isn't that. This is not that. This is a completely different thing. Okay. Okay. If you're confused about how this is not related to pea glow, then please email me or speak up now or forever hold your peace. Please, because I don't want to get into why this isn't pea glow unless you think that it is. Okay. All right, because it's not. If the antibody sees the antigen on the slide, the antibody being linked to something that glows, now you can see the slide. If your patient doesn't have the antigen, the antibody won't stick and it won't glow. That's immunofluorescence. I feel like it's a pretty simple one. Doesn't require all these enzymes and substrates like ELISA does. Okay, Western blot is like even more specific than ELISA is. Let's talk a little bit about Western blot. In Western blot, you would take a gel. Here's a well in the gel, and you'll put your sample, your protein, your nasal swab fluid, or whatever. Now, instead of putting drops of your nasal swab fluid in little tiny wells and mixing it with antibody and seeing if it cleaves and causes a color change. And then measuring that color change. How dark is it? Means you have more antibody, whatever. Instead of doing that, we're going to run it out on a gel and separate out whatever else is in that mess, because there could be all sorts of proteins in your patient's sample. It's not just the antigen, right? And to avoid it interfering with other proteins in the sample, we'll separate out the proteins by electrophoresis. Okay, so we'll run an electric current through it, and it works the same way as the DNA one does too. The smaller ones at the bottom, the larger ones at the top. And you would expect your antigen to be a certain size because you're a scientist and you know. So if your antigen is supposed to be this size, we would take all of this out of the gel and use more electricity to transfer it to a paper. From the gel to the paper. Then we'll move that. Here's your paper now. And you have all these proteins on there. And you expect your protein, your antigen, to be this size. Now, do I want this color? If your antibody binds here, and we'll label it with something. If your antibody binds there and it's the same size, it's like double proving that it is your antigen and you are positive for it. So if somebody pops positive on an ELISA for having antibodies to HIV, we might do the Western blot to confirm that it really is HIV antibodies. So we can do that with the Western blot and actually prove that this patient has those antibodies to HIV. So this is something we use as a confirmatory, very, very specific and very, very sensitive, even more than ELISA. So if we got a positive ELISA, we will usually confirm with Western blot so that we are not making any mistakes in calling somebody positive, just because proteins got mixed up and reacted a certain way. We don't want to risk that if we're calling somebody HIV positive, for example. Another test that we have to be aware of in our immunologic type tests is the complement tests, and it relies on complement lysing red blood cells if the patient has antigen. Well, usually we look for antibody with. I'm just trying to get rid of all this stuff. Again, it's based on the immune response. So we're looking at antibody and antigen. So if we have a well, I hate this one, I hate explaining this one because it gets so confusing. If we have a well and your patient has antibody to a known antigen, then your patient will bind with that antigen. And the complement that's hanging out in there won't interact with the antigen. Normally, if it interacts with the antigen, then red blood cells that are in there will get lysed. But in this case, they won't. So it's whether the red blood cells get lysed or not by complement that determines if your patient has the antibody or not. So it's like a multi-layer thing. This is a test that is used in labs, but I feel like this is just a multi-level test that has a lot of levels in it that I feel like we're not quite ready for. The reason that I'm bringing it up this way is that what I want you to know for the exam is not how the complement test functions as an immunologic test, step by step, and why it's positive and not. What I do need you to know, though, is that it is an immunologic test. Okay? But yes, okay. It is based on antigen recognition. That's what you need to know. That's your take-home message. All right, moving on to the tuberculin skin test. If you have T cells that recognize tuberculosis, and we inject you with tuberculin from tuberculosis, and you react to it within 2 to 3 days, you'll get a welt on the skin. Then that would mean that you've seen it before, and that this is a test that's done in vivo, which just means in the living body. Okay. Any questions about the immunologic test before we move on to the genotypic tests? We'll do genotypic in red. Nope, not what I meant to do. All right, genotypic tests, genetic or genotypic, okay. Genetic or genotypic tests are PCR, which is polymerase chain reaction, which is amplification of DNA sequences. This relies on the primer binding to the sequence. If the primer, which is specific, binds to the DNA sequence, then we can elongate. If the primer doesn't bind because the sequence isn't there, then it can't elongate. And the machine measures if it's elongating or not. If it elongates, you're positive. If it doesn't, you're negative. So that's how PCR works as a diagnostic test. PCR falls into a category known as nucleic acid amplification testing, NAAT. You'll often see NAAT in your case studies, okay? But PCR is a type of that. We also have nucleic acid hybridization. This is using a probe, I don't know, who cares, T, C, G, whatever. And it's linked to a fluorescent molecule. Again, it'll glow if it binds. And here we have your patient's swab sample or a biopsy, any type of sample, their stool sample spread out on a slide, whatever. If that patient's swab or stool or whatever has the sequence T, 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 A, G, C, then these will bind to it and stick and cause it to glow. If it glows, it's positive. If it doesn't, it's negative for the gene sequence. You see how this looks exactly the same as our immunofluorescence, but our immunofluorescence depended on antibody or antigen reaction. This depends on nucleic acid sequence, but it's a similar concept. If it glows, it's positive. If it doesn't, it's negative. This, where we have nucleic acid hybridization, but this is fluorescent. This would also be considered in situ, okay, which is kind of like in vitro, but it's in situ. Because I can just take your patient's swab sample and then treat it right there without having to do all these different steps in an in vitro test. So in situ hybridization. That's what that is, a depiction of. FISH. It's literally called FISH. Even in scientific communities, we will call this FISH. Okay, so that's what FISH is. It is a type of genotypic test. All right. Then there's microarrays. Microarrays use this type of ideology, except instead of just like taking one type of antigen and seeing if it matches to one type of antigen, in a microarray, we're taking a plate. We're putting several probe markers on it, and the several probe markers might associate with different types of genetic sequences or different types of mutations or whatever, but all of them genetically related. It's all genotypic. This is all DNA-based sequence. And then put your patient's sample all over it. And if it binds to certain spots, they'll light up more. And then that would be indicative of having those specific types of mutations. And you might have to have multiple of them to verify that this is in fact this disease or that disease or whatever. So that's what a microarray is. You can also do whole genome sequencing, and that might be useful if we have a new type of pathogen or if we have some sort of new antibiotic resistance or something like that to learn more about the pathogen, sequence its whole genome. So those are all possibilities in identifying pathogens. Our last group is our other group. So these are our others. All right, mass spectrometry. And a specific type of mass spectrometry called MALDI-TOF. TOF in MALDI-TOF refers to time of flight. And the way that this works is you have a plate with a sample on it, and we hit it with a laser. and it causes particles to fly off. I'm not even drawing the particles very well. And when they hit a detector, whatever, it says detector, I promise, then it signals to the computer. And the time that it takes for the molecule to fly to the detector, the time of flight, determines like the mass or even the charge of the particle. And this helps you break down the exact molecular makeup of whatever substance is on the plate, the slide, whatever it is, okay? Whatever substance you're testing. So you can get the exact molecular makeup of it. So this can help you identify pathogens or drugs or all sorts of stuff that you might be dealing with in your patient. It's very, very useful. It depends on physics and chemistry. And it's not, I can't read the results myself, but the computer can. And that's what I can tell you about it. Do we have any questions? We know imaging stuff too, right? CT scans, MRIs, PET scans, X-rays can be useful in identifying things as well. So if we're looking for a tuberculosis infection, we might do an acid fast stain and an X-ray just to confirm that all of the things are there for the diagnostic process. Okay. Questions about the content? So we're going to go into the actual like practice stuff for our most missed concepts next. All right, most missed concepts. We're going to start with chapter 17, which was our immune pathologies, disorders of the immune system. First, we have our type 1 hypersensitivities. Remember that type 1 is mediated by IgE. We consider it to be an immediate hypersensitivity because once you see the allergen, it pretty much causes a reaction almost immediately within minutes. It's not delayed in any way. So once you have your initial sensitization, then it should be every time after that, you have a reaction within minutes. Common triggers, we have our contactants, our inhalants, our injectants, and all of that, which we covered previously. Requires sensitization, re-exposure, leading to degranulation of the mast cells, leading to histamine and other inflammatory mediators, leading to hives and itching, swelling of the skin. We have respiratory difficulty breathing, basically bronchoconstriction, and whatever results from that. We have gastrointestinal issues that can result, as well as cardiovascular stuff in the case of our anaphylactic response, drop in blood pressure, dizziness, even fainting, due to actual literal blood loss from the brain as a result of the shock response associated with this. We can use antihistamines or corticosteroids for atopic local stuff, but for systemic treatment, we would want to reach for epinephrine for an immediate effect. Best way to prevent it is through avoidance or desensitization when it's available for certain allergens for that. All right. So here are some example questions for us. Which of the following is the most immediate and effective treatment for anaphylaxis? So remember, if we are having atopic reactions, we would reach for our steroids or our diphenhydramine antihistamines, that sort of stuff. So we won't want oral antihistamines for anaphylaxis. We want it for atopic. Nor will we want intravenous corticosteroids. Again, that would be for atopic. You can also use inhaled bronchodilators and corticosteroids for atopic, like asthma. But for anaphylaxis, you want epinephrine injections. It could be intramuscular or it could be intravenous. When we are sensitizing for type 1, what role does IgE play in the response? Does IgE neutralize the allergen in the bloodstream? Does it bind to the allergen receptors on mast cells or basophils? We didn't talk much about the basophils' role, but it has the same role. This should be our answer B. Does IgE activate complement proteins? That would be a type 2 reaction, type 2 hypersensitivity. Do they enhance phagocytosis of the allergen by macrophages? That's a process called opsonization, which is a function of some types of antibodies, but that's not what mediates the type 1 response. In the type 1 response, it's that degranulation of the mast cells or basophils. So B. All righty, next question, or next topic. The hemolytic disease of the newborn. Again, this is usually associated with Rh incompatibility. Again, the mama being a negative and seeing the positive baby as foreign and making antibodies, which upon labor could cause death of the newborn. There's other factors that can be involved. So that would be the case of the Rh incompatibility. But you can also have hemolytic disease of the newborn from ABO compatibility. It's actually more common, but it's less concerning. And so in this case, it wouldn't be positive and negative, that would be the issue. It would be like if the mama was an O and the baby was an A. A would look foreign to an O. And that would be the case with ABO. With the ABO stuff, you're more likely to have things like jaundice, some anemia, maybe an enlarged liver or spleen, and maybe a situation of hydrops fetalis, where this would be the fetal buildup of fluid in the fetal tissues from the attack, which can be potentially life-threatening. This whole reaction would be most severe with Rh group incompatibility. So when the mama was a negative and the baby was a positive, that's the most severe with that. We could still have jaundice and anemia with ABO rejection, but it's not as life-threatening. The risk isn't as high for that. RhoGAM basically interferes with the Rh stuff. So RhoGAM is where you have Rho or D, immune globulin, Rho, like the rhesus factor, immune globulin, where we administer that to Rh negative mamas during and after pregnancy, typically, to prevent the formation of Rh antibodies. So we usually give this to any Rh negative mama just to prevent her from making these antibodies and then blocking that whole response that way. So it prevents that reaction from happening, so that we don't have that likelihood of rejecting a baby in that situation. It protects the baby that you're having, as well as getting any of that sensitization for future babies as well. So it's useful. We usually administer RhoGAM for anybody that we test as Rh negative if there's any reason to think she could have a positive baby. So there's that. Okay, cool. What do I say about this? I think that's fine. Okay, we're going to go on to the next slide. So question about this one. Which of the following is the primary method to prevent hemolytic disease of the newborn? If you know that the mom is going to be a negative and the baby's going to be potentially be a positive, what's the best way to prevent the reaction there? And that's going to be administering RhoGAM. Routine blood transfusions during pregnancy won't prevent the attack. Again, if the mama is a negative, we can't transfuse her with positive to desensitize her. That'd kill the mama. It wouldn't work. So not routine blood transfusions. Providing high-dose vitamin K won't prevent this. Vitamin K is not going to prevent hemolytic disease, not prevent it. All right, using antiviral medications during pregnancy, this is not an infection, so using antivirals won't prevent anything dealing with that Rh rejection situation. All right, next question. A newborn is diagnosed with severe jaundice, anemia, and an enlarged liver shortly after birth. The mama is an AB negative. If mama is AB negative, A looks normal to mama, B looks normal to mama, and O looks more normal to mama. Have a feeling, yep, the baby's going to be positive. This is not ABO compatibility. This is Rh compatibility. The baby has hemolytic disease of the newborn because of the Rh group here. So why is this being caused? Is it from Rh incompatibility leading to hemolytic disease? Probably, but what were our other choices? ABO incompatibility? No, the ABO group was compatible. Is this from hepatitis? No, we know this is the most likely indicator. The mama saw the baby as foreign. Maternal diabetes, this is not the issue either. It's going to be the Rh incompatibility. All right. I also have tissue grafts on here. I put 3 on here, so it's just 2. Okay, so for this is just reviewing those different tissue graft types in a little bit more detail. Again, autograft is self to self. Isograft is genetically identical twins. Very, very minimal risk for rejection there. Allograft is from a person who is not an identical twin or self. And the xenograft is from a non-human animal. So pig heart valves and other situations where we might use pig skin or animal skin for transplant in severe burn situations where there's not enough for autograft situations. So let's move on to our question about this stuff. So again, auto, self. Iso, twin. Allo, other person. Xeno, animal. All right, so which type of tissue graft is most likely to require lifelong immunosuppressive therapy to prevent rejection? So which one would be the least compatible? Would be xenograft. Xenograft would be the most likely. But we also, I would want to say, probably less likely. Well, it depends on the tissue that we're transplanting. It could be D or C. This would not be a good question for the exam because it could be D or C, both of these. Just because it just depends on the tissue and it depends on what you're doing. So transplants between genetically different individuals, you're likely to reject, leading to lifelong immunosuppression. Depending on the tissue from the xenograft. So if we're doing certain xenografts that we reach for, like heart valves and things like that, due to the nature of that type of the tissue, which is not like a whole organ, but it's a specific type of like connective tissue or something like that, because of the nature of the tissue, you're just naturally less likely to reject it. So it depends on the type of tissue. So it could be C or D in this, depending on the type of tissue. Traditional transplants that are done, like heart and liver and lung transplants. All right. We're not doing xeno transplants with those. We're doing allografts. And in that case, C would be. So it just depends on the tissue and the type of transplant there to answer that effectively. The second question, a patient receives a heart valve transplant from a pig. Which type of tissue graft is this? And what is a major concern with this? Xenograft, right? Because autograft would be from self. Isograft would be from a twin. Allograft would be from another human. That's not a twin, not genetically identical. Okay. Now, self would have very low risk of, what would be a concern associated with a self graft? Well, you wouldn't reject a self graft. There's very little concern with this. An isograft, also very little concern. Almost no to low risk of rejection. Allograft, you would need immunosuppressive drugs, but in a xenograft, you have a high risk of rejection and potential disease transmission from the animal, depending on the tissue, where it came from, and all sorts of stuff like that. Just depends. All right. Chapter 18, stuff. How about if I actually click on the thing? All right, phenotypic testing. We talked about a lot of different phenotypic tests, the biochemical tests, selective and differential media, staining. All of those are phenotypic. We talked about the phage typing, and we talked about a little bit about Kirby-Bauer. So some bacteria are naturally resistant to certain antibiotics, and that can help with identifying the bacteria as well. Not just useful in choosing how to treat your patient, but also potentially in helping diagnose the infection. So those are all phenotypic tests. Immunologic tests rely on antigen recognition. Genotypic or genetic diagnostic tests rely on actual genetic sequence, ATCG code. All right. Let's see if we can get into it. There's more info on this slide if you feel like reading more into it, but we're going to try to get into that question. So, phenotypic, a lot of them are biochemical in nature, also includes our phage typing and our Kirby-Bauer. Immunologic, these are going to rely on antibody and or antigen. And then genotypic, is going to rely on the actual ATC DNA sequence. All right, so first question, which type of diagnostic test would be most appropriate for identifying a bacterial pathogen based on its genetic material? So this question, oh, you're not even seeing the same thing as me. Here it is. There you go. This question is basically asking us, which of these tests is a genotypic test? Well, you tell me. Is gram stain genotypic? It is phenotypic. ELISA is immunologic. Polymerase chain reaction. Now we're cooking with fire. Biochemical test strip, phenotypic. So our answer is C. That's the one that's depending on the genetic material. Genotypic test. All right, a patient undergoes a test to determine the presence of antibodies against HIV. What type of diagnostic test is being used? And we're looking for antibodies and antigen. That's going to be an immunologic test, which is B. Not phenotypic, which depends on basically like biochemical aspects of the organism, descriptive aspects of the organism. Not genetic, which depends on the genetic sequence. And a biochemical test is a type of phenotypic. Okay. Right, our last topic is sensitivity and specificity. Of course it is. Of course it is. It's our favorite. We know, but we have to keep perspective on this, okay? Sensitivity, more sensitive equals less false negatives. More specific equals less false positives. And again, oh, right, it would help if I showed you the slide. And again, yes, I just saw your post over here, Jordan. I'm just reading this xenograph story. There's a guy from hometown that had a full pig heart transplant when he was in 8th grade. Yeah, that's wild. That's definitely a xenograft. Definitely. It's something they've experimented with a lot. There's a high risk of rejection with stuff like that, but not for everybody, not for everybody. And it depends too, how old you are. Can your body still train your body to call that self? And so that the younger you are, the more likely your body can train your body to not react to a thing for stuff like that. So anyways, back to what we're talking about here. Specific and sensitive. I want to look at this. So if you're very sensitive to something like the princess and the pea, right? Okay, you know the story of the princess and the pea, I hope. That the princess laid on a mattress and she felt the pea under her mattress. So she put like 20 other mattresses up there over the course of the story. And she could still feel the pea under 20 different mattresses on top of the pea. She was very sensitive. She felt even one pea, even through 20 mattresses. It's wild stuff, but she was very sensitive. So she was not likely to call there not being a pea, which would be a false negative. The pea was there. She was so sensitive that she still recognized the pea. The pea was still there. And if she were to say, oh, I don't feel it anymore, even though it was still there, that would be a false negative. That's what a false negative is. To say something is negative, but it's actually positive. The pea is still there. I wouldn't feel a pea under 20 mattresses, but she did. That's the idea of the princess and the pea. I don't know. It works for me. Maybe it works for you. Whereas specificity. If the princess were laying on a mattress and she were to say, that's a pea under my mattress, but it was actually a rock, then she's not very specific. Okay, so that's the idea. That would be a false positive. If she were laying on that mattress and she says, I feel that pea under there, but it's not a pea, it's something else, it's a rock, then that would be a false positive. False positive. Okay, so specificity is being specific about what the issue is. Sensitivity is being able to identify even the smallest amount of the issue. All right, so let's see if we can get into the question about it. I'm going to write a little note out to the side for it. Specificity. If you are specific, you will have low false positives. If you are sensitive, you will have low false negatives. And again, this is just pointing out what they pointed out on our last time when we were reviewing the P and the N. P for positive, N for negative. So whatever helps you with it, that was a good catch. I think it was Deontay that pointed it out the first time, but yeah, it's a really good one. Or maybe it might have been Deontay or it was Maria. I don't remember who, but I want to give credit to you guys for that. Anyways, diagnostic test that has a sensitivity of 95%. What would that mean? Again, sensitivity is dealing with false negatives. So if you are 95% sensitive, that means out of 100 people that are positive, you will correctly identify that 95 of them were positive with the test. And you would say 5 were negative, even though they're positive. Are we tracking that? Okay. That's what that's saying. Say, let's see our choices and walk through them. The test correctly identifies 95% of the patients without the disease? No. It's 95% of the patients have, again, 100% of them have it, and you're only identifying 95% of them as positive. That's going to be sensitivity. Next choice. The test correctly identifies 95% of the patients with the disease. Yes. That's, so far I'm with this one. C, the test has a 95% chance of being positive in healthy individuals. No. If you're healthy and we're dealing with sensitivity, then if you're 95% sensitive, then we shouldn't be marking you as having the disease. Okay. Saying that you're positive in a healthy individual will be a false positive. That's a specificity issue, not a sensitivity issue. The test has a 95% chance of correctly diagnosing the disease when it is absent. Oh, no. How would you correctly diagnose a disease when it is absent? That doesn't even make sense. So our choice is B. A new test for tuberculosis is designed to have high specificity. Should have a low false positive. What is the primary benefit of this characteristic? What is the benefit of having a high specificity? That you will have low incidence of false positives. So let's see what our choice is. It's for tuberculosis. The test will detect a high percentage of people with TB. It might detect a high percentage of people with TB, but that's not dealing with the sensitivity. That's dealing with more of, it's not dealing with sensitivity or specificity. The test will accurately confirm that a person does not have TB if the test result is negative. Okay. That is dealing with, yeah, this is where the wording, and this is where I hate the wording, because explaining this out to you guys, it makes sense in my mind, but how do I explain this out? All right, so if somebody doesn't have TB and you say that they are negative, this is where people get tripped up. So how do I divide this out in my line of thinking? So this is where people get tripped up, is they'll be like, oh, it talks about negative, false negative, that's sensitivity. Not always if we're talking about negatives and positive. I'm talking about a false negative, that's sensitivity. But specificity, now we can still be talking about negatives, but we're not talking about false negatives. Okay. So if the test accurately confirms that a person does not have TB, that means that the people that have TB, we're also accurately identifying them. Okay, so this could be the answer. Okay, okay. If it's negative, we mean that it's negative. It's very specific, okay? Specificity here's the issue, okay? Specificity and sensitivity. We're specific for something. We're calling something negative specifically. I know. Okay, let's read through our other choices though. The test will have a high rate of false negatives. Again, if we're talking about specificity, we should have a low rate of false positives. Okay, low rate of false positives. If you have a high rate of false negatives, that would be a low sensitivity. So it shouldn't be C. The test will identify a high number of non-TB infections. If it's highly specific, we shouldn't be identifying things other than TB. So if you're very specific, you will know the difference between positive TB and negative TB. You will not call something that looks like TB positive or something that doesn't look like TB negative, or that you can know that if we're talking about TB, we're talking about TB because we are specific for TB. So if I call you negative for TB, I mean you're negative for TB, not for something that looks like TB, or positive for TB, not something that looks like TB. Keeping it specific. So B is our answer. I hate the wording on it. And I promise I won't make the wording this tricky on the test, but B is our answer because it's accurate about what is specifically being tested. Okay. As opposed to A, which would be sensitivity. I know. I can already feel it in your bones from this far away. What questions do we have? Does anybody have any questions? All right, if you guys don't have any other questions, that is it for today's review. I appreciate you guys joining in. Email me if you do get questions. Yeah, thank you for showing up. I'll post the recording today. Otherwise, I hope to see you guys at 4 p.m. on Sunday for our Kahoot review, or feel free to play through the Kahoot on your own time. Happy studying, and I will see you guys for the exam on Monday or Tuesday. Thank you guys.